All right, I don't know what happened to the last video, but we're going to try it again. We're going to talk about conduction of electrical signals by neurons. In the nervous system of animals, specialized cells called neurons form a complex network that receives, processes, and transmits information from one part of the body to another. The center of this network is located in the brain, which stores and analyzes information and transmits signals to different parts of the body. The nervous system is very complex, with about 10 to the 10 interconnected neurons. The messages transmitted by neurons are voltage pulses called action potentials. When a neuron receives a strong enough stimulus, it produces identical voltage pulses that are actively propagated along its structure. The stronger the stimulus, the more pulses are produced. When the pulses reach the end of the neuron, they activate either muscle cells or other neurons. There is a firing threshold for neurons. Action potentials propagate along a neuron only if the stimulus is sufficiently strong. Neurons can be divided into three classes: sensory neurons, motor neurons, and interneurons. The sensory neurons receive stimuli from sensory organs that monitor the external and internal environment of the body. Depending on their specialized functions, the sensory neurons convey messages about factors such as light, temperature, pressure, muscle tension, and odor to higher centers in the nervous system. The motor neurons carry messages that control the muscle cells. Those messages are based on the information provided by the sensory neurons and by the brain. The interneurons transmit information from one neuron to another. Each neuron consists of a cell body with attached input ends called dendrites and a long tail called the axon, which transmits signals away from the cell, is shown in the figure on the left. The far end of the axon branches into nerve endings that transmit signals across small gaps to other neurons or to muscle cells. A simple sensory-motor neuron circuit is shown in the figure on the right. A stimulus from a muscle produces nerve impulses that travel to the spine. Here, the signal is transmitted to a motor neuron, which in turn sends impulses to control the muscle. Okay, this is just a figure that shows an electron microscope image of neurons in the brain. All right. The axon, which is an extension of the neuron cell, conducts electric impulses away from the cell body. Some axons are extremely long. In humans, for example, the axons connecting the spine with the fingers and toes are more than 1 meter long. The neuron can transmit messages because the axon has special active electrical characteristics. The axon acts as an active source of energy, like a battery. Shown is an experimental setup to probe the electrical and chemical properties on an axon. Note that the outside of the axon is grounded, so all measured voltages are with respect to a zero potential on the outside. The probes can be used to inject current into the axon, measuring the resulting action potential as a function of time at a fixed point, and sample the cell's chemical composition. Experiments like these are usually difficult to run because the diameter of most axons is very small. Even the largest axons in the human nervous system have a diameter of only about 20 times 10 to the minus 4 centimeters. The giant squid, on the other hand, has an axon with a diameter of about 0.5 millimeter, which is large enough for the convenient insertion of probes. Much of the information about signal transmission in the nervous system has come from experiments with the squid axon. In the aqueous environment of the body, salts and other molecules dissociate into positive and negative ions. As a result, body fluids are relatively good conductors of electricity. The inside of the axon is filled with an ionic fluid that is separated from the surrounding body fluid by a thin membrane that is only about 5 nanometers to 10 nanometers thick. The resistivities of the internal and external fluids are about the same, but their chemical compositions are substantially different. The external fluid is similar to seawater. Its ionic solutes are mostly positive sodium ions and negative chloride ions. Inside the axon, the positive ions are mostly potassium ions, and negative ions are mostly large organic ions. Ordinarily, the concentrations of sodium and potassium ions inside and outside the axon would be equalized by diffusion. But the axon is a living cell with an energy supply and can change the permeability of its membrane on a timescale of milliseconds. When the axon is not conducting an electric pulse, the axon membrane is highly permeable to potassium ions, slightly permeable to sodium ions, and impermeable to large organic ions. This means that although sodium ions cannot easily enter the axon, potassium ions can leave it. As the potassium ions leave the axon, they leave behind large negative organic ions, which cannot follow them through the membrane. As a result, a negative potential builds up inside the axon with respect to the outside. The final negative potential reached, which has been measured to about 270 millivolts, holds back the outflow of potassium ions so that at equilibrium, the concentration of ions remains. The mechanism for the production of an electric signal by the neuron is conceptually simple, but was experimentally difficult to sort out. When a neuron changes its resting potential because of an appropriate stimulus, the properties of its membrane change locally. As a result, there is a sudden flow of sodium ions into the cell that lasts for about 2 milliseconds. This flow produces the 130 millivolt peak in the action potential shown in the top figure. Immediately afterward, there is an increase in potassium ion flow out of the cell that restores the resting action potential to 270 millivolts in an additional 3 milliseconds. Both the sodium and potassium ion flows have been measured by using radioactive sodium and potassium tracers. The nerve signal has been measured to travel along the axon at speeds from 50 meters per second to about 150 meters per second. This flow of charged particles or signal transmission in a nerve axon is unlike signal transmission in the metal wire. In an axon, charges move perpendicular to the direction of travel of the nerve signal, and the signal moves much more slowly than a voltage pulse traveling along a metallic wire. Even though the axon is a highly complex structure, and much of how sodium and potassium ion channels open and close is not understood, standard electric circuit concepts of current and capacitance can be used to analyze axons. The axon, having equal and opposite charges separated by thin dielectric membrane, acts like a capacitor. This ends the section, and this also ends the chapter. All right.